Every single physical movement that you make begins as a lightning-fast electrical command, originating straight from the human brain. While the experience of thinking feels abstract, the mechanics that translate a thought into a muscle flex are strictly physical. Every thought is a measurable event, driven by a sequence of precise, predictable mathematics. In the 1950s, scientists Alan Hodgkin and Andrew Huxley set out to measure the electrical signal of a firing neuron. At the time, human nerve cells were far too microscopic for their sensors to record. They looked for a biological subject large enough to study with the naked eye, eventually turning their attention to the ocean. They chose the giant squid. It possesses a single nerve fiber so thick that researchers could physically thread a metal wire through its center. This setup allowed them to capture the raw electrical pulse of an active nerve, visualizing the data as a distinct wave on an oscilloscope. This biological quirk gave Hodgkin and Huxley the first direct look at the electrical mechanics of a thought, allowing them to translate living tissue into calculable data. At rest, a neuron maintains a negative electrical charge, the resting potential, at exactly negative 70 millivolts. Positive sodium ions mass outside the cell, while positive potassium ions remain inside. Voltage-gated ion channels act as gatekeepers, preventing them from crossing the barrier. This separation of ions creates potential energy. The cell sits in a state of constant tension, waiting for an electrical trigger to release a flood. To model this behavior, Hodgkin and Huxley moved past the biology and began treating the cell as a hardwired electrical circuit. The cell membrane, which stores an electrical charge, functions exactly like a capacitor. Those ion channels function as variable resistors, regulating the flow of electrical current through the circuit by opening or closing. Mapping the neuron onto an engineering schematic allowed researchers to apply rigorous calculus, predicting how a nerve cell would react to any given stimulus. The researchers had to account for how wide these channels would open. They needed to calculate the exact probability of ion flow based on the current voltage. They introduced three gating variables, N, M, and H. These probabilities between 0 and 1 determine whether a specific channel is open or closed. The potassium current relies on 4 gates opening in unison, expressed mathematically as n to the 4th power. Sodium channels use a fast-acting activation gate, M, followed immediately by an inactivation gate, H, that stops the flow. These three probabilistic variables form the underlying code for the speed and duration of every single human thought. When the cell reaches a specific electrical threshold, the sodium gates snap open. Positive ions rush into the cell, shifting the internal charge toward positive. This produces a dramatic upward spike on a voltage graph, representing the moment a neuron fires. Almost instantly, the H gate slams shut and the potassium channels open, flushing positive ions out of the cell and returning the voltage to its negative baseline. This sequence, the rapid spike and the immediate recovery, is the action potential. It is the physical manifestation of a single human thought. By combining these variables, Hodgkin and Huxley produced a single differential equation that tracks how the total current through the membrane changes over time. This equation accurately predicts the shape of the action potential graph, providing a mathematical framework for the entire nervous system. Every time you recall a memory or coordinate a movement, millions of neurons across your brain are solving these exact calculus problems in real time. The brain functions as a biological supercomputer, flawlessly executing these complex equations using only meat, salt, and electricity.